are in listen-only mode.
Hello and welcome to today's webinar. We will be covering Advaita's new application, iVariantGuide. My name is Andrew Olson. I'm the Vice President for Business Development here at Advaita. And today we have one of hopefully many upcoming webinars on our new variant application. Today's presenter is Dr. Cordelia Zeraldo. Cordelia has a background in computational biology with expertise in systems biology approaches to understanding inflammation and immunology. She will be going over an overview of our new application, iVariantGuide. And just as a reminder, if you have any questions along the way, please use the dialog box in the GoToWebinar control panel to ask your questions. We will be taking them at the end of the webinar. And also at the end of the webinar, we have a very short 3-question survey that we would really appreciate if you could fill out. That'll also help guide future webinars and topics in the future. So with that, I'd like to turn it over to Dr. Cordelia Zeraldo. Take it away, Cordelia.
Thanks, Andrew. Hi, everybody, and welcome. Today, I'll introduce Advaita's brand new variant analysis application, iVariantGuide. It's so new that it's still in beta, which means that for now it's completely free to use. I hope that by the time we're done today, you will feel confident enough to try iVariantGuide with your own data and see its power for yourself. Just briefly, today I'm going to tell you a bit about the company, give you some background on variant analysis, talk about the benefits of iVariantGuide, and then show you the application. I'll focus on how to upload data, use the interactive filters, and get the most out of your data, and then why modeling variants on impacted pathways and associated gene ontology terms gets you to more efficient hypothesis generation. I'll show you how to share reports with collaborators without any additional subscriptions, and we'll leave time for questions at the end. First of all, who is Advaita? Our team is made up of a small number of people, but is very strong. We're a group of bright, dedicated, energetic people from diverse backgrounds. And Advaita happily partners with many organizations, some of which are shown here. Our mission is to help you make use of your next-gen sequencing data. So our tools are designed to help you focus on the biology. We do that by implementing the most advanced computational methods so that you get accurate results. We maintain a comprehensive knowledge base that we upgrade regularly. We just finished an update last week so that you don't miss anything. And we work hard to make sure our applications are easy to navigate so that you can focus on your data instead of getting lost in the software. It's easy to find variants of interest using iVariantGuide. We annotate your variants comprehensively so that you have all of the information you need. Our interactive filters help you see connections between features in your data. And Advaita allows you to model your variants on pathways and associated gene ontology terms to explore the biological context. In addition to our powerful functionalities, iVariantGuide has several features to enhance your experience. I'll just mention a couple. Our processing time is quick. It only takes a few minutes to fully annotate your data set, which means you can have answers within the hour. We also have online sharing, which means you can collaborate with anyone, anywhere, anytime. Our secure cloud-based platform means you can both view your data at your own convenience, wherever you are in the world. We also offer custom filter presets, which can be used across data sets. This saves you time and also makes your analyses automatically reproducible. Some of our earliest users have said very complimentary things about iVariantGuide. They often mention how comprehensive the application is, providing multiple analyses in a single tool They mentioned the time savings this brings by not having to import data into multiple tools. Others have also commented on the speed of processing data. The worst is when you set your data to process right before you leave for the evening and then come back in the morning only to find that it has failed overnight and you have to start all over. Even at high traffic times, we keep the upload and processing to less than half an hour, generally. Before we can annotate them, you must identify the variants in your sample. And this begins with sequencing of whole genome, whole exome, or sequencing gene panels. Once the reads come off a sequencer, they are aligned to generate a consensus sequence. And this is what's compared to a reference genome assembly by the variant calling algorithm. All of the places that differ from the reference are listed in a variant call file, which is the input for variant analysis. The problem is that for a single sample, VCFs often contain hundreds of thousands of variants, which if you're trying to organize using a spreadsheet program can feel like wrangling a fire hose. That's where iVariantGuide comes in. We wrangle the VCF so that you can quickly filter your hundreds of thousands of variants into a short list of candidates of causal variants. So here's what an iVariantGuide workflow might look like. You first add your samples from your VCF and check their stats. Then you use those samples to generate a report in the same page where you have added your samples. Once you click Create Report, we start annotating your data, and when it's ready, the status bar will turn green on the Reports page. Next, you can enter your report and view the statistics, filter your variants, view details, and save presets. You can view details of any specific variant by clicking on its record in the table and viewing its clinical significance, linking out to ClinVar, as well as associated pathways and gene ontology terms. Once you're satisfied with your filtered set of variants, you can explore their biological context in pathways and gene ontologies. And finally, when you're done, you can create a printable report to save your analyses and publish your results. So let's take a closer look at just a few of those steps. iVariantGuide accepts samples in the form of VCF files, version 4.1 or later. There are a couple of size limitations. In order to ensure that the interface retains its snappiness and your interactive filtering experience is satisfactory, we take VCFs smaller than 100 megabytes or zipped VCFs smaller than 20 megabytes. Once your samples are uploaded, you can use them to create a report, and you will receive an email when it's done annotating just a few minutes later. Meanwhile, in the back end, iVariantGuide is busy bringing together a wide variety of information so that you can analyze your data. We annotate at multiple levels of biology, from sequence level information to transcript and gene level predicted impacts, to cellular and molecular functions, all the way up to clinical significance. And we provide links out to additional sources so that it's easy to find anything you need. Then, we make it easy for you to take advantage of all of that rich information with our custom filters. Within a handful of clicks, you can filter your entire variant list down to a few variants of interest. In this example, we first eliminate false positives by setting lower thresholds on quality and read depth. Then, since we are looking for novel variants, we exclude those found in ClinVar and other databases. Finally, to identify deleterious mutations, we choose variants predicted to have high or moderate impact on the transcripts they affect. In this example dataset, this leaves us with about 300 novel deleterious mutations. We can save this filter combination as a preset to use again on this data set or any other we choose. We also recognize that there's almost an infinite number of ways to combine filters to identify causal variants of interest. On the same data set, we can apply a different set of filters to identify disease-associated deleterious mutations. In this case, we find 16 candidate mutations in our data set. And this is where most other applications stop. But we're not finished with variant analysis because we don't have an actionable hypothesis to take back to the bench. In iVariantGuide, you can model your variant set on associated pathways and get a systems biology view of your variant data. Identifying relevant pathways is already a huge step beyond just having a list of candidate variants, but then we allow you to view your variants along with their predicted impact directly on the pathway map. You can even overlay information about microRNAs and drugs that target genes on the pathway, allowing you a powerful view of tools you might use to test the variant's predicted effect on that pathway. There are a number of ways this can be used for hypothesis generation, and the important point is that with this analysis you can generate testable predictions about your subjects. In the upcoming release, we will offer a comprehensive printable report that may be used to publish your analysis results in a manuscript. This will include all of the analysis available in iVariantGuide, a full table of variants in your filtered set, graphs of how the features break out across your set, variant details, as well as pathways and gene ontology terms analysis, detailed methods, and references. Now let's take a closer look at iVariantGuide. Okay, this is our login screen. I'm just going to open the demo account. Okay, you will land on your reports page. Each of these lines is a single report that has been created that we can explore. But before we explore, I want to show you how to upload data. In the top right, there's a plus sign which you click that will take you to our samples page. On the right-hand side is the dialog for creating a new report, which you will fill in by selecting a sample from the left. Your available samples are on the left. You can add a title and description and then click Create. If your sample is not available yet and you need to upload it, click the blue plus sign. Select the reference assembly for your new file and then browse to select your file. Once you've done that, you can click the Upload button. And once the file is first uploaded, before we start annotation, we provide you summary stats, which are also available here. I think it's a good idea to look at your summary stats every time you upload a sample because this is sort of a sanity check. Make sure you selected the correct reference assembly. It'll let you know if we found anything odd in the file. Once you're satisfied, you can add your sample to the available samples, and then you can use that to create a new report. So I have this sample here selected, and you can see in very light gray the name has also transferred to the right-hand side. Once I create a title and a description for this report, I can choose Analyze Data to begin annotating. I've already done that, so I won't go through that now. We'll just go back to the reports page. Here's that created report on the bottom row, and I can explore either by clicking this link icon or opening the details and clicking Explore in the right-hand panel. Okay. Once our data loads in, you'll see that this is your main variance page where you have your entire unfiltered data set and all of your filters and information is here. Of course, it's slow when everybody's watching. This file is from an RNA-seq experiment. mRNA was taken from a breast cancer patient, and this sample was used to compare different types of breast cancer. We're going to look at what's in this single sample today. As you can see below the variance table, the total number of variants in this file, unfiltered, is 34,363. And we can view the statistics of that file by opening these interactive filters, which double as graphs reporting what's happening in your variant set. You can see, for example, that the majority of the variants in this data set are SNPs, but there are also some deletions and insertions. Hovering over a pie section will tell you how much it contributes to the graph. You can also click any segment to filter. So for example, if I click SNP, then what I've done is I've filtered the variants to only include the SNPs in the data set, which takes out about 1,500 variants from the data set. We also provide a search bar that can be used as a way to filter as well. You can type the name of a disease or the name or an official gene symbol to search for variants associated with either of those things that are in the data set. So if I type the word cancer, what I get are variants associated with diseases that have the word cancer in the name. I can also add additional terms. If I type malig, maybe the disease doesn't have the word cancer, but it has the word malignant. This will add a few more variants to our data set. So we're at 26 total variants that are associated with diseases that contain either cancer or malig. And you can see that every time I change a search filter, these graphs update automatically and dynamically, which allows us to see associations that we wouldn't have seen if we had just filled in all of our search criteria at once. For example, in the chromosome pie, you can see that chromosome 5 takes a much larger portion than it did when we had all of the variants available. So that's interesting that it's overrepresented among disease-associated variants. If you click it and filter to only variants in chromosome 5, you get a map of the chromosome. And in fact, you can select a region of the chromosome by, again, clicking and dragging this window to include your region of interest. So we're down to 5 filtered variants that are associated with the terms in the search bar and located in this region of chromosome 5. Clicking on any record in the table, for example, this one at the bottom, opens up the variant details view. And here is where you get all of the information about this specific variant, starting with the chromosome position and allele change. Here next is the dbSNP ID as well as a link out to the dbSNP record. We list all of the ways that this specific allele change is annotated and the predicted transcript impact. Next is the clinical significance section. So here, this variant is associated to cancer progression and tumor cell motility in ClinVar. And each of these is a separate link to a ClinVar accession, and you can link out and view all of the associated information. In many cases, there are also links to other databases with more information about the linked disease, such as OMIM, Orphanet, SNOMED, Gene Reviews, and a few others. Below that, pathways and gene ontology terms that are associated with genes impacted by this specific variant are listed. And you can click on any one of these now, or I recommend we set a preset before we do that. So all of this information is specific to this variant, and you can see it for any variant in your data. Returning to the filters, I want to show you some interactive filtering by drop-down menu. You can choose multiple selections by clicking the title of a graph and then choosing what you want. Each of these graphs is exportable. So if you click the arrow here, we generate an image that you can download as either a PNG or SVG. And every single graph on the page has that option. Once you're satisfied with your filters, you will probably want to save them as a preset. And you do that by clicking the Save As button in the upper left, entering a name for your filter, and clicking Save. Your presets are located in the left-hand sidebar. Here you can see the list of filters that the list of presets that I have previously saved. And here, second to the bottom, is actually the same set of filters that we described earlier in the presentation to identify novel deleterious mutations. So predicted high or moderate impact, minimum depth 4 and quality 20, no clinical significance recorded, a very rare allele frequency, or not available, meaning not recorded. So if I select this preset, it will clear all of my previous selections. I select the right one. And all of my graphs update accordingly. If you remember, I said we had about 300 novel deleterious mutations in this data set. And you can see that below the variant table, in fact, we have 293. So now that we have a preset selected, we can explore the biological context in the Pathways page. The first thing you'll see when the Pathways page loads is a list of pathways that are associated with genes associated with variants in your filtered set. And on the right-hand side, there's a number of genes on each pathway impacted by variants in your set. They're color-coded by the predicted impact level, that transcript impact that we were talking about earlier. And they're ranked by the number of impacted genes. So I'm going to select the PI3 kinase signaling pathway. And what that does is it opens up pathway details, starting with a description of the pathway and then the diagram, which you can zoom and scroll to your region of interest. I'm going to zoom in so that it's easier to see. As you can see, we have high impact variants affecting genes on the pathway that are at the very beginning. So we might expect to have quite a bit of impact on this pathway. Hovering over any one of the nodes reveals all of the genes that are involved in that node, and you can select any number of them to further filter your variant set to only those genes. But as it is, the variant set is listed below the pathway map. And you can also view microRNAs and drugs that target genes on the pathway. So if I select a microRNA, you'll see that when I scroll up, let's select a couple of them. The targets of that microRNA are circled on the pathway in light blue. Same for drugs. If I select a drug, the targets of that drug will be circled in orange on the pathway. This, as you can see, starts to give you some context in terms of what are the types of experiments you could do to test the effects of the variant on this pathway. The other thing that's nice, I think, is that we have a specific set of filters applied right now, but it's true that there are other variants in the sample that might be impacting this pathway. So we can change the preset while we're in the pathway's view, which I'll do by opening the preset tab on the left. And I'm going to just choose this false positives filter. So this is the first step that gets us in that filtering cascade that gets us from 34,000 down to 14,000. And I just want to take away any variant that might just be an artifact of the data processing. So anything that we believe is real, no other filters applied. When we do that, you can see many more genes on the pathway light up. And you can start to see a little bit broader view of the mechanisms that might be affected by your data. When you're done exploring pathways, you can export this pathway map, and you have control over what section of the map you export, whether it's all of it or just a certain region, same as the viewing. And then it exports as either a PNG or a vector graphic, just like the other graphics in the program. Okay, that's sort of how pathways work. Once you're satisfied with your analysis, you can go back out to the reports page. Perhaps you're extremely excited about something you found and you want to show a colleague. The way to do that is to click this share link next to the record, which opens up a sharing dialog. And you can share in one of two ways. We allow you to enable a public link. Right now I have one enabled already. If I click disable, it will take it away and create a new one once I click enable. You can copy this link and use it for anyone who wants to access this data. If you would like to share it in a more private mode, you can enter an email. So by doing this, only the person who receives the email will have ability to access your data, and you can control whether or not that person can forward the link to anyone else. If you unclick allow to reshare, then that link is only good for a single user to access. In this way, you can control who sees your data online, but give them full access if you choose. Okay, I think that's it. I hope I convinced you today that iVariantGuide gives you powerful variance analysis in an easy-to-use package. Our cloud platform is secure and annotation engine is fast, meaning you can do analysis anytime, anywhere. Our interactive filters make your data easy to navigate and help uncover hidden associations. Our powerful pathway analysis allows you to generate new hypotheses about your data and get meaning out of your variants. And the ability to share interactive reports means that your collaborators can explore the data as much as they want, increasing the amount of insight you can get from the same analysis. I'm happy to take any questions you have. Please use the chat boxes to submit them. And while those first questions come in, I want to remind you that iVariantGuide is completely free to use while it's in beta. We're planning to release the full version next quarter, and any reports you create now will always be yours to keep. I'm happy to schedule personalized demos. Just call or email and we'll set up a time. And check your email soon for announcements of upcoming webinars. Our gene expression pathway analysis application we're going to show how to analyze time series data in iPathwayGuide soon.
Okay, great. Thank you, Cordelia. So we do have a few questions that are coming in here. Again, if you do have a question, please use the dialog box. I see they're starting to come in. Also, at the end of the webinar, if you wouldn't mind, please taking a 3-question survey. That'll really help us to make more informative and better webinars for you in the future. So with that, let's take some of these questions. The first one we have is, are genomes restricted to human only?
Currently, we offer HG19 and HG38 and no other organisms, but we are open to suggestions, so if there's something you desperately want, please reach out.
All right, great. Let's see, we have actually this is a 2-parter, it looks like. Will the presentation be published online to watch later? I can actually answer that one. Yes, I've failed to mention we are recording this webinar and we will post a link. Actually, we'll post it on YouTube. We'll put it on our support page for iVariantGuide. But you will, if by attending this or having signed up for this webinar, you will also get a link automatically. The second part of the question is there a possibility to have this software run locally? I guess as opposed to cloud-based.
Yeah, currently our implementation is cloud-based so that we can take advantage of sharing and access.
Right. Yeah. Actually, I do have a little bit of additional insight on that. I know certain customers have asked if they could maybe have it run in their own private instance in a cloud-based system. That is a possibility. If that's something that you desperately need, please reach out to us. We would love to work with you on setting up something like that. Okay. Next question we have is, what is the source of the pathway content?
So all of the sources of all of the annotations and data are available in the FAQ section of our website. But I can tell you that pathways come from KEG.
Right, and KEG stands for the Kyoto Encyclopedia of Genes and Genomes, I believe. So that's a good question. All right. Excuse me here. Do you assess gain or loss of function?
So that's what our predicted impact score does. Generally, we're looking at loss of function mutations there.
Okay.
I
okay.
believe.
Here's an interesting question. I think it's a good one. Do you have an alternative palette available for colorblind customers?
That's a great question, and I wish the answer was yes. But again, it's a good feedback. That's something that we can work on.
Right, and while it's in beta, we appreciate any kind of questions or comments about that. We'll definitely take those into consideration and see if we can't include those in the commercial release. What is the source of clinical significance? Only ClinVar?
That's right.
Okay, so at this point, it's only ClinVar that we're using, right?
Yeah, we offer links out to those other databases, however, so that you get more complete information that way.
And I know you kind of covered this, but just to reiterate, here's a question here. What's the max size of a file that you can upload?
So max size would be a VCF that's not zipped at 100 megabytes. Regardless of size, we recommend that you keep the number of variants in your file below 200,000. Again, this is to make sure that the filters work in the way that they're supposed to, meaning as quickly as they're supposed to, so that you get that immediate feedback that's so critical.
Gotcha. And I think we have time for one more question here. And by the way, if we didn't get to your question, we will answer it after the webinar concludes. We'll follow up with you individually. But the last one that we have here is, can we import other annotation sources into iVariant Guide? The
answer to that question is not yet. It's something that many people have asked about and we've thought about. In this first release, we're limited to only the sources that we provide. However, we do realize that offering personalized annotations would open up a whole world of new possibilities. So it's something that we are thinking about and figuring out if we can incorporate.
Very good. Okay, so I think with those questions answered, we will conclude today's webinar. Again, please take the survey. We'd really appreciate it. And if we didn't get to your question, we will definitely follow up with you in the next day or so. And finally, one last plug. If you happen to be in the East Coast, particularly in Boston, we are going to be at the Festival of Genomics in Boston next week. It's a free conference, so if you can get yourself there, you can attend. It's on Tuesday and Wednesday at the Boston Convention Center. I think the times are like from 8 a.m. to 5 or 6 p.m., but you can look, just Google Festival of Genomics Boston, and you'll see it. We'll have a booth there, and any specific questions you may have, just come on by, and we'd love to meet you and answer any questions you may have there. So thank you, everybody, and have a great rest of your day.
Thanks.